Good afternoon. So this article is skeletal stem cells, skeletal stem and progenitor cells maintain cranial suture, patency and prevent craniosynostosis. So this was published in the Nature Communications, Impact Factor 15.7. The authors are from Stanford, USA, and following terms you should know. Cranial sutures, it is fibrous joint between the skull bones, allow cranial growth, and premature fusion of this will lead to craniosynostosis. It is a congenital condition where one or more sutures fuse too early, causing skull deformities. The posterior frontal suture, it is the suture in mice. This study uses that to study the physiologically fusion in early life. The patent suture, it is sutures, example sagittal, coronal, lamboid, that remain open and maintain growth potential into adulthood. The skeletal stem cell progenitor cells, so it is CD51 plus and CD200 plus. It is a defined stem cell population found in sutures, characterized by surface markers CD51, CD200 with multipotency and self-renewal ability. The canonical CWNT signaling, it is a key signaling pathway that maintains stem cell populations. Activation promotes suture patency, inhibition induces fusion. The axin 2, it is a negative regulator of WNT signaling. The axin 2 LAGZ plus mice have enhanced WNT activation. So used in this study for rescue of craniosynostosis. The twist1 mouse model, it is a genetic model of syndromic craniosynostosis caused by the twist1 haploinsufficiency. The SB431524, it is a TGF beta pathway inhibitor in experiments to mimic non-syndromic craniosynostosis by reducing SMAD6 expression. The FACS, it is fluorescent activated cell sorting. It is a technique used to isolate specific cell population. Here they did for CD51, CD200 progenitor cells. The bulk RNA sequencing, it is transcriptomic profiling method that measures average gene expression in a cell population, whereas the single cell RNA sequencing is high resolution sequencing to identify transcriptional heterogeneity in individual cells. The pentachrome staining, it is a histological method to visualize the tissue architecture including suture, mesenchyme and bone formation. The suturectomy, it is surgical removal, a standard treatment for craniosynostosis. Here they did in mice. And the resynostosis, the refusion of the surgically corrected suture, it is a frequent complication after suturectomy. The EDU incorporation assay, it is an experimental method to measure the cell proliferation by labeling newly synthesized DNA, and clonal analysis. It is a genetic labeling method to trace proliferation and lineage of single progenitor cells in vivo. So the novelty of this study, it is the study examines the cranial suture biology through the lens of the skeletal stems, morphogenital progenitor cells, which had not previously been categorized in this context. So it demonstrates that equilibrium of these cells is linked to the suture patency and provides a new cellular perspective on craniosynostosis. So the authors hypothesized that the transplanting skeletal progenitor stem cells together with WNT3A protein following a surgical ablation of synostotic suture could recreate a physiological suture mesenchyme and prevent the suture refusion. So they wanted to use the stem cells to recreate the sutures in the fused suture after the surgery. They aim to investigate the cranial suture biology and craniosynostosis pathogenesis by profiling, isolating, and functionally testing skeletal stem cells within the suture mesenchyme. They first had to objectives were they first had to isolate and characterize the skeletal stem cells and from cranial sutures. Then they had to compare the dynamics between the patent and fusing sutures. They had to investigate the role of canonical WNT signaling in regulating these cells, and they had to test whether the cells with WNT3A transplantation could prevent the recurring synostosis after suturectomy. So in materials, the animal, they used multiple mouse strains. These were the strains and the models for normal and craniosynostotic sutures. The FACS isolation. So they isolated the CD cells with CD51 plus and CD200 plus markers using fluorescence activated cell sorting, and the bulk RNA sequencing they did, which where they profiled the global gene expression difference in progenitor cells from patents versus fusing sutures. They did single cell RNA sequencing, where they examined the transcriptional heterogeneity and subpopulation dynamics within the suture derived progenitor cells. They did RT-PCR validation to confirm the expression changes of selected craniosynostosis associated genes, and the ex vivo suture explant culture they did, where they cultured the dissected sutures with inhibitors to mimic non-syndromic craniosynostosis. The WNT activation and inhibition, where they applied WNT3A activator or DKK1 or SFRP1 for inhibitors to test the WNT signaling effects on cell frequency and suture patency. The differentiation assay, they assessed the multipotency of progenitor cells by inducing osteogenic and chondrogenic lineages. The colony forming and self-renewal assay, where they evaluated the capacity of isolated progenitor cells to proliferate and maintain stemness. The histology and immunofluorescence, they performed pentachrome staining and beta-catenin immunostaining to visualize tissue architecture and WNT activity. And lastly, they did the in vivo suturectomy with transplantation, where they surgically removed synostotic sutures and transplanted it with CD51 stem cells without and with WNT3A signaling to prevent to test the prevention of refusion. So the statistical analysis, they did compared to two-day students t-test. One-way ANOVA for group comparisons. They used the software graph pad Prism 8, and the significance was greater than equal to 0.05, and the sample size was not predetermined. Sorry, less than. And the sample size was not predetermined. The results from the least two independent experiments per condition was considered. So the results, the in vivo profiling of the stem cells. So these skeletal stem cells were conformed in cranial sutures and decreased sharply in the physiology and fusing posterior frontal suture. So they were less in number there, and remained stable in patent sutures. The transcriptomic signaling of sutures derived cells, so bulk and single cell RNA sequencing revealed distinct transcriptional programs with PF suture cells showing down regulation of craniosynostosis related genes, marking them for fusion. In the imbalance of cell and craniosynostosis, both syndromic and non-syndromic craniosynostosis models showed significant decrease in the stem cells, progenitor cells, compared to normal sutures. In the modulation of WNT signaling, the activation of canonical WNT increased the progenitor cells, and preserved the suture patency, while inhibition of WNT decreased them and induced pathological fusion. The activation of WNT rescue craniosynostosis. So in these strains of mice, the WNT activation restored the stem cells level, skeletal stem cells level, and prevented coronal suture fusion and maintained the patency similar to the wild type. Lastly, the preventing re-synostosis after suturectomy. The transplant of these cells after along with the WNT3A signaling after suture ablation prevented suture refusion, while either treatment alone was ineffective.